Good afternoon, everyone. The circulatory and lymphatic systems are essential components of the human body. They are responsible for the transport of vital substances, which would include oxygen, nutrients, hormones, cells, all throughout the body. Understanding the microscopic structure of the circulatory system is crucial for comprehending their complex functions and the processes that occur within them. The circulatory system is composed of the heart, blood vessels, and blood cells, all of which work together to maintain proper blood flow and oxygen delivery throughout the body. The heart, a muscular organ, pumps blood through a network of blood vessels, which includes arteries, veins, and capillaries. Arteries contain or carry oxygen-rich blood away from the heart to the body's tissues, while veins transport oxygen-poor blood back to the heart. Capillaries, which are the smallest blood vessels, facilitate the exchange of nutrients, oxygen and waste products between the blood and surrounding tissues. Blood cells, which include WBCs, RBCs, and platelets, are critical components of the circulatory system, and they perform a wide variety of functions, such as the carrying of oxygen, fighting infections, and facilitating of blood clotting. The lymphatic system, on the other hand, is responsible for maintaining fluid balance within the body and protection against infections. It is composed of lymphatic vessels, lymph nodes, and lymphoid organs, such as the thymus and the spleen. Lymphatic vessels carry a clear fluid, which we call lymph, and this contains immune cells, proteins, and waste products away from the body's tissues and towards the lymph nodes, where immune cells can identify and attack any harmful pathogens present. Studying the histology of the circulatory and lymphatic systems is important for several reasons. First, it provides a deeper understanding of the structure and function of these systems and how they work together to maintain homeostasis throughout the body. Secondly, our understanding aids in the diagnosis and treatment of various medical conditions such as heart disease, lymphedema, and leukemia. Histologic examinations of tissue samples can reveal abnormalities or changes in the structure of the circulatory system and the lymphatic system, which can indicate the presence of disease or disorders. Finally, understanding the histology of these systems is essential for medical professionals such as surgeons and pathologists who may encounter these issues during procedures or if they need to interpret biopsy results. As an introduction, the histology of the circulatory and lymphatic systems is an important area of study for anyone interested in anatomy and physiology, as it provides a foundation for understanding the complex functions of these systems and how they contribute to overall health and well-being. So the circulatory system primarily serves to deliver oxygenated blood to cells and tissues and to return venous blood to the lungs for gas exchange. The major components of the circulatory system is the heart, the arteries, the veins, and the capillaries. Okay? The main function of the circulatory system is transport of oxygen and nutrients to the tissues. In doing so, they also concurrently carry away carbon dioxide and waste products from the tissues. They also circulate hormones from their sites of synthesis to their target cells. And blood flow to the surfaces of the body serve to regulate body temperature. At the core of the circulatory system is the heart, which, as mentioned, is a muscular pump that propels blood at high pressure around the body throughout the blood vessels. The heart is a four-chambered muscular organ composed of two atria that serve to receive blood from the systemic circulation and from the pulmonary circulation, and two ventricles, which pump blood to the lungs and to the peripheral organs. The heart is surrounded by a fiber sac, which is called the pericardium. Okay? The pericardium is composed of multiple layers, as which we will be talking about later. The heart is composed of multiple layers which parallel the layers of the blood vessels. Okay, so these cardiac layers are called tunicae. Okay, and you have, and from the inside going out, the layers are the endocardium, which is analogous to the tunica intima of the blood vessels, myocardium, which is analogous to the tunica media of blood vessels, and the epicardium, which is analogous to tunica adventitia. The heart also contains a fibroskeleton, heart valves, which separate the various chambers of the heart, and within the layers of the heart, you also have a system of um, specialized conductive fibers, which serve to generate and transmit impulses to the heart to facilitate effective contraction. Now, the endocardium is the layer that lines the lumen of the heart. It is the innermost layer and is composed of simple squamous epithelium, similar to that of the tunica intima. It is underlined by what is known as the subendocardium, which is a layer of loose connective tissue with interspersed adipose tissue. Okay, so this is the endocardium here at the bottom. The loose layer immediately above the endocardium is the subendocardium. Okay? The thickest layer of the heart is the myocardium, and this parallels the tunica media of blood vessels. It is composed of cardiac muscles, which, as we have talked about last time, is composed of branching striated muscles with the cells connected by intercalated discs that contain gap junctions that facilitate easy transmission of electrolytes necessary for the propagation of um, electrical impulses. The myocardium is important because its contraction provides the strength necessary to propel blood into the arteries for distribution to the peripheral tissues. The central support of the heart is provided for by the fibroskeleton. This is composed of thick bundles of collagen that is oriented in various directions. It is composed of the septum membranaceum, the trigona fibrosa, and the annuli fibrosi. Okay? The fibroskeleton, the fibroskeleton supports the valves, and it does this by preventing overstretching of the valves um, as blood rushes into the various chambers. Okay? The fibroskeleton also serves as the insertion point of the cardiac muscle fibers and serves as an electrical insulator between the atria and the ventricles. The heart valves, on the other hand, are composed of a skeleton of fibrous connective tissue, which is continuous with the fibrous system of the heart. It allows blood to flow only in one direction through the chambers because as blood rushes through the next chamber, the backflow causes the valves to close. Okay? It is also lined on both sides by endothelium and are attached to the annuli fibrosi. Okay? So um, the valves include the atrioventricular valves, which are the tricuspid valve between the right atrium and the right ventricle, and the mitral valve between the left atrium and the left ventricle. You also have the aortic valve, which separates the left ventricle from the aorta, and the pulmonary valve, which separates the right ventricle to the pulmonary artery. Crucial to the proper functioning of the heart, or the proper contraction of the heart, is the impulse generating and the impulse conducting system. This is composed of of a number of specialized nodes and bundles of fibers that facilitate the transmission of impulses in the heart. This is important because um, synchronized contraction of the heart is necessary for um, proper propagation of blood, for the generation of enough force to propel blood throughout the body. Okay, so the main pacemaker of the heart is known as the sinoatrial node, and the SA node is responsible for the generation of impulses that initiate contraction of the atrium. Okay, the AV node is similar in structure to the sinoatrial node, um, and in some cases can become the primary pacemaker of the heart if the sinoatrial node is damaged. Okay. The AV bundle of His, located here, is a band of conducting tissue radiating from the AV node into the interventricular septum, where it divides into two branches. This structure is important because um, the impulses should only course between the AV node and the atrioventricular bundle of His. This is the reason why the fibroskeleton, and the reason behind this is that um, the the fibroskeleton acts as an insulator which prevents the generation of impulses from the atrial muscle cells to the ventricular cells. And finally, we have Purkinje fibers, which are large modified cardiac muscle cells. Arteries, on the other hand, conduct oxygenated blood away from the heart to the capillaries. And we have four types. We have the large elastic arteries, which are also known as conducting arteries. They receive blood directly from the heart, and as their name suggests, they are elastic. They're capable of stretching to accommodate the massive amounts of blood and the strong pressure from emanating from the heart. The main example of an elastic artery is the aorta, as well as its major branches. You also have smaller, medium-sized arteries, known as distributing arteries. They are also known as muscular arteries because their tunica media tend to be thicker. They distribute blood to various organs and are named after, and distributing arteries have specific names depending on where they are in the body. Emanating from the muscular arteries are even smaller arteries, which we call arterioles. They are the smallest arteries and are responsible for regulating blood pressure in the body. Arterioles give rise to smaller branches, and we call them metarterioles. Metarterioles then give rise to capillaries, where the nutrients and important gases diffuse to the tissues. As with the heart, the artery is composed of three basic layers: the tunica intima, which is the innermost layer. Similar to the subendocardium, it is composed of simple squamous epithelium. Underneath the simple squamous epithelium is subendothelium, which is composed of connective tissue, particularly collagen. And outside the external to the subendothelium, you have an internal elastic cartilage. Okay? The tunica intima is important because it is a dynamic structure in that it prevents clotting when it prevents spontaneous clotting of blood in normal in vivo conditions, but when it is needed, it has a lot of procoagulant molecules that facilitate clotting when there is a break in the integrity of the tunica intima. The simple squamous epithelium or the endothelium secretes a lot of anticoagulant substances like heparin, which prevents blood from clotting spontaneously. We know that spontaneous clotting of blood is disastrous, as they can form blockages in the smaller blood vessels in the body. However, when there is a breach in the endothelium, This facilitates the exposure of a lot of molecules in the subendothelium, particularly collagen, von Willebrand factor, that um, have a high affinity towards the circulating platelets, and this will start the cascade of blood clot formation. Okay, so there is a tenuous balance between anticoagulant and procoagulant activity in the blood, and what determines whether the milieu or the internal environment of the blood vessels is anticoagulant or procoagulant is facilitated by the tunica intima. Now, external to the intima is a smooth muscle, is a thick layer of smooth muscle, which is known as the tunica media. It is said to be the thickest portion of the artery and corresponds to the myocardium of the heart. Externally, external to the tunica media is the tunica adventitia, and this represents the outermost layer. It is composed of a simple squamous epithelium, which is separated from the rest of the artery by a basement membrane. Coursing through the adventitia is loose connective tissue, blood vessels, and occasionally nerves and smooth muscle cells. The tunica adventitia has an external elastic membrane, and in larger arteries, um, within the tunica adventitia are smaller blood vessels that supply the bigger arteries, and we call these small blood vessels vasa vasorum. Okay. In contrast, the veins conduct blood away from the organs and tissues to the heart, so they carry deoxygenated blood back to the heart for delivery to the lungs. Okay. As with the arteries, veins can be large and small and medium. Okay. The largest veins in the body include the vena cava, the pulmonary veins, and the mesenteric veins. You also have emanating from the large veins are the medium and small vessels, and these would include the jugular vessels and the other named veins of the body. Venules, on the other hand, are the smallest and are involved in exchange of metabolites and diapedesis. Diapedesis is the movement of um, white blood cells from the periphery of the blood vessels into the through the walls of the veins into the tissues. Okay, we see this in inflammation, and and this represents the white blood cells going to the sites of inflammation. Okay, now the veins are also composed of three layers with similar names: tunica intima, media, and adventitia. Okay, they're lined by the same um, lining epithelia and same components. So the tunica intima is composed of an endothelium. Okay. However, um, in veins there is no internal elastic layer, and there is no subendothelium. Okay. So this is the reason why atherosclerotic events only occur in arteries, because it is the arteries that have subendothelial procoagulant molecules. The tunica media is also composed of smooth muscles, but in comparison to arteries where the tunica media is really thick, in the tunica media of veins are very thin. Okay? In veins, what is the thickest layer is the tunica adventitia. They also contain vasa vasora, but they do not have an external elastic layer. Okay, so this is a comparison between an artery and a vein. Okay, the tunica media is very thick in the artery, but is thin in veins. Okay, the adventitia is thicker in veins and is very thin in arteries. Okay. Now, capillaries are a single layer of endothelial cells. Okay, they're composed of simple squamous epithelium, and this facilitates transfer of nutrients from the blood into the tissues where they are needed. They exhibit selective permeability because they facilitate the entry and exit of substances um, through the capillaries while preventing the movement of other molecules. Okay, because they are only composed of a single layer or a single layer of endothelial cells, they do not have tunica media and tunica adventitia. Okay? There are different types of capillaries, and these include continuous capillaries, fenestrated capillaries, and sinusoidal capillaries. Continuous capillaries are the most common, and they contain pinocytic vesicles, except in the CNS. They do not have holes or fenestra, and the basal lamina is continuous. Okay? They are located in nervous tissue, muscles, connective tissue, exocrine glands, and the lungs. Okay, so they don't have holes. They do not have fenestrae. Okay, so the lining is continuous. Fenestrated capillaries, on the other hand, are characterized by openings which we call fenestrae. Okay, because they have holes, they facilitate rapid exchange of molecules between blood and the tissues, and we see this in the choroid plexus, the small intestines, the kidney, the glomeruli of the kidney. Okay, and then finally we have sinusoidal capillaries, which are irregular and winding or tortuous. Okay, they have very wide diameters, and because the diameters are quite wide, these facilitate in slowing down of blood. Okay, basement membrane is either incomplete or absent, which allows the direct exchange of molecules. And we see these specialized capillaries in organs like the liver, the spleen, and the bone marrow. Onto the lymphatic system. The lymphatic system is part of the immune system. Now, the lymphatic system is composed of the immune system working hand in hand with the various lymphoid organs in the body. The immune system consists of a large, diverse population of cells in the blood called WBCs, which interact, um, develop, and mature in various organs in the body called lymphoid organs. And these lymphoid organs are interconnected only by the blood and the lymphatic circulation. There is no physical connection between these lymphoid organs. Now, immunity can either be innate or adaptive. Innate immunity is non-specific. This means that regardless of what type of organism it is, the body reacts the same way. It involves a variety of um, interacting mechanisms, and the response for innate immunity is usually innate. Because regardless, as I've mentioned, regardless of the organism, the response is the same. There is no priming involved. There is no recognition of the specific type of organism or foreign body involved. Innate immunity can be because of physical barriers in the body or by cells circulating certain types of circulating WBCs. Now, physical barriers include the skin and the mucous membranes of the gastrointestinal tract, the respiratory tract, and the urogenital tract. And they not only prevent infection or penetration of the host body, but these surfaces may have antibacterial or anti-organismal substances that um, that kill these organisms. Um, in some areas of the body, they also have resident flora or resident bacterial flora, um, which compete with pathogenic organisms for nutrition and for nutrients. And in doing so, they prevent the colonization or growth of pathogenic organisms in these areas. Um, Innate immunity can also involve a majority of WBCs in the blood. These include neutrophils, eosinophils, basophils, and monocytes. Okay, so these WBCs act the same way, um, initiate the same pathways regardless of the organism that it meets. In contrast, adaptive immunity is mediated by lymphocytes and antigen presenting cells. Um, it takes time for them to respond because these cells need to be primed. They need to uh, have prior sensitization before they can mount an immune response that is specific to whatever organism it is. Um, the adaptive immunity is a response to specific microbial invaders and involve production of memory B cells. As you can see here, most of the types of WBCs fall under innate immunity, while B cells and T cells tend to fall under adaptive immunity. Okay, B cells work by secretion of antibodies. Okay, um, specific organisms are recognized by um, antibodies that these organisms are specific to. And these antibodies, these detecting antibodies, are usually located on the surface of B cells. Now, once these antibodies interact with the antigen that it is specific for, um, that B cell undergoes clonal proliferation. It multiplies in number. Some of these cells become memory B cells, which hide in certain parts of the body for them to be ready for um, subsequent infections of this specific type of organism. Other B cells mature into um, plasma cells, which are mature B cells that secrete the same antibody that it is specific for. And these antibodies are produced in massive amounts and they serve to neutralize um, that specific organism, or they also facilitate opsonization or phagocytosis. Okay. Now, cytokines are a wide variety of chemicals in the body, and they act as communication chemicals of the cells in the immune system to coordinate defensive measures. They are both innate and they fall under both innate and adaptive immunity. Okay, they mediate chemotaxis, which is the movement of um, WBCs towards the site of inflammation, and they also mediate diapedesis, which is the movement of white blood cells from the blood to the tissue space. Cytokines also stimulate increased mitotic activity, and as mentioned, they stimulate phagocytosis or direct cell killing. A few slides ago, I mentioned uh, about the fact that specific antibodies react with specific antigens. Now, when we talk about antigens, they are molecules that are recognized by cells of the adaptive immune system and which elicits a response from these cells. They are usually proteins and polysaccharides because um, these molecules are large enough and varied enough to produce a wide variety of antigenic sites. They are structurally varied. Okay, antigens may are usually proteins from intact cells, like bacteria, protozoa, and tumor cells. Antibodies are large globular proteins, and they follow this more or less the same structure. Y-shaped structure consisting of two heavy chains and two light chains. Okay, we have five types of antibodies: IgG, IgM, IgA, IgD, and IgE. The most abundant of which is IgG. This is the most abundant and is increased during infections. They are highly soluble and stable and are capable of crossing the placental barrier into the fetal circulation. It is responsible for passive immunity against certain infections until the newborn's own adaptive immunity is acquired. IgM antibody is a pentamer in that it still has it still follows the same Y structure, but are usually grouped into five of these. Um, this is the first antibody to be produced in response to an antigen and is the most effective antibody class in activating the complement system. The complement system is a series of proteins that bind to a foreign cell, which leads to the destruction of that cell. IgM and IgG are capable of binding complement. IgA is an antibody that is present in secretions. Okay, in contrast to IgG, which usually occur in the monomeric form, and IgM, which occur in the pentameric form, IgA occurs in the dimeric form. Okay, and the two Y structures are joined together by a structure known as the joining chain or the J chain. The IgA antibody is produced by plasma cells in a mucosal membrane, so digestive tract, respiratory tract, and the urogenital tract. The IgA molecule also has a secretory component which protects the antibodies from proteolysis by enzymes in the secretions. Now, IgE is found on the surface of mast cells and basophils, and they facilitate the release of histamine, heparin, and leukotrienes. This is particularly abundant in allergic reactions and parasitic infections. Now, complement activation involves the participation of a group of proteins around, there are around 20 types of these proteins that are produced in the liver. So these are known as complement proteins. Okay, they are activated through a series of chemical reactions, with one reaction leading to the next. So this is known as a cascade of enzymatic reactions. Once complement is activated, specific complement components bind and rupture membranes of invading cells. They also clump antigen-bearing bacteria or cells and facilitate chemotaxis. Now, one of the effects of complement activation is opsonization, and this refers to the ability of receptors on macrophages, neutrophils, and eosinophils to recognize and bind the Fc portions of antibodies attached to surface antigens or microorganisms. Now, we have a certain subset of immune cells known as NK cells or natural killer cells, and um, they are responsible for detection of um, antibodies bound to antigens on virally infected cells. So once the NK cells are activated, they release certain enzymes called granzymes and a molecule called perforin, which destroys the virally infected cell. This was discussed in a previous lecture, but um, T cells are also part of the adaptive immune system. However, they differ from B cells, whose recognition relies on the presence of antibodies bound on the membranes of the B cells. T cells recognize um, specific proteins through the interaction with a phagocytic cell with phagocytic cells known as antigen presenting cells. So, as mentioned a while back, antigen presenting cells gobble up um, foreign microorganisms and partially digest these organisms. Now, these partially digested proteins will be complexed with a molecule known as major histocompatibility complex, or MHC. Now, MHC, together with these peptide fragments, will be expressed into the cell membrane of the antigen presenting cell, which the T cells will then be able to recognize. And then the T cells will then produce the cytokines necessary to mount an immune response. So precursors of lymphocytes are located in the bone marrow. However, um, they as part of their maturation process, they leave the bone marrow. Some of them leave the bone marrow to go to the thymus to mature into T cells. Another subset remain and differentiate in the bone marrow to become the B cells. Okay, so this is a screenshot of the lymphoid organs. You have the thymus gland. Okay, the superficial, it is covered by a capsule. Here you have the cortex and you have the medulla in the center. Lymph nodes. For lymph nodes, you have again a capsule, a space underneath known as the subcapsular sinus. The superficial portion is known as the cortex, the inner portion is known as the medulla. In the cortex, you see a lot of lymphoid follicles that mature through different stages. Okay. So that ends our brief discussion on the circulatory and immune system. As usual, if you have any questions, kindly post them on our Telegram group. Thank you very much.